All right, this is going to be a short video that's going to visually illustrate some of these concepts that we read about in Jensen's chapter 2 and chapter 3 of the teenage brain. Namely, we're going to show you some pictures and I'm going to give you a little bit more of an explanation about how some of these things like action potentials, long-term potentiation, dendritic branching, and synaptic pruning look in real life, because understanding the mechanisms that are responsible for this typical brain development and just general neuroplasticity is going to be really important for understanding some of the applications and implications that we talk about later on when it comes to adolescence, like poor connectivity, um, imbalances of connectivity, and stuff like that. So this is just a general picture. It's not a real picture. It's a illustration of what a very sparse neural network might look like. But first, let's get down to the nitty-gritty of what a neuron might look like. And some of you might have learned this in biology or even an intro psychology course. But this is a single neuron, okay? This is the actual cell body, which we would call the nucleus. And for our purposes in this class and really in psychology, that's the least important part of the neuron for you to know. The important parts of the neuron are going to be the ones that help it communicate and make connections with other neurons. So starting on this end over here, we have the dendrites, and these are essentially like tree branches, okay? So they come out of the cell body and they're meant to catch things that are coming from other neurons. Then we have the axon and the myelin sheath, which we discussed or we read about in the book. This axon is essentially the long cord that the electrical signal that a neuron generates travels through. And then at the end, we see these other long, they're called axon terminals. They're other long fingers similar to dendrites. And these are going to be what spits neurotransmitters out to other cells. So on this side, we receive information from other cells, and on this side, we spit information out to other cells. And on both sides, we have synapses at the end of all of these, okay? And this is, I couldn't fit enough in this picture, but there could be hundreds and hundreds of synapses on each one of these, okay? So just know that up close, both of these sides house synapses that have receptors or shoot out information. So let's look at an action potential, which we read about. And this is the basic mechanism of how neurons communicate with each other. So like we said, over here, remember these are the dendrites. There would be other neurons around this, and these other neurons are going to be releasing neurotransmitters, okay? These neurotransmitters are going to come into this neuron, and if they are excitatory, like we read about in the book, they're going to excite the neuron and potentially create an action potential. But if they are inhibitory neurotransmitters, like a painkiller you would take as an inhibitory transmitter, because when it fits into the receptors of a neuron, it keeps the neuron off. And so a neuron would be one of the things that makes you feel pain. It sends impulses and signals to your nerves. And so when we turn that neuron off, it doesn't send those signals. But if we have a excitatory neurotransmitter that's being accepted into this neuron, it's going to excite the neuron. You have to have enough of it to get it excited enough to release that action potential, which is literally like a small electric jolt that's going to go down the axon right here. And remember that this axon is coated in a myelin sheath. And so this myelin sheath is literally, it's lipids, it's just fat. And so it essentially keeps that electrical signal from seeping out of the axon so that almost all of the energy gets down to the end here where these axon terminals are. Now remember, each of these axon terminals is covered with synapses. And so what we're going to look at next is a picture of these two, of a close-up of this right here, which would be the way that these two neurons are going to communicate with each other. So this is a close-up of what we were just looking at. This is a synapse at the end of an axon terminal. In it, we have these vesicles that are loaded with neurotransmitters, and each neuron is going to have a different neurotransmitter. It might be inhibitory or excitatory, and there are various hundreds actually types of within each types of those, but either way, it has whatever kind of neurotransmitter that it carries, And after an action potential, the action potential is going to induce those neurotransmitters to be released. Okay, and they get released into what we call the synaptic cleft, which essentially is just the empty space that's between two synapses. They never directly touch. It's always going to be this empty space in between them, but they're very close together. On the other side, so this is at the end of a dendrite, so on the receiving end of another neuron, it has all these little black guys right here are receptors. Okay, the receptors are housed on dendrites, and they are specially fit to accept certain neurons. So Jensen used the analogy of a key into a keyhole. So assuming that these receptors fit this neurotransmitter, they will kind of plug into this neurotransmitter here. Now, going back to the concept before, if this is an excitatory neurotransmitter like dopamine, and enough of these neurotransmitters get expelled and then accepted by these receptors, it's going to create the whole process. It's a chain reaction. At this point, it's going to excite this neuron, and if it excites it enough, it will then elicit an action potential in that neuron, and the chain continues. So this is the basic mechanism of how cells communicate with each other. But how do cells change according to experience? So one of those, the concepts that we read about in the book is long-term potentiation, okay? Long-term potentiation, if you were to go Google it, it's going to be very complex because it has to do with a number of chemicals that are signaling changes inside these synapses. But the main idea of it is if this neuron is continuously releasing being activated and releasing neurotransmitters to this neuron, then these neurons have a reason to form a stronger bond. So what they do is they start to produce the neuron that's sending the neurotransmitters out produces more neurotransmitters in that synapse, and it will also grow more synapses. And this receiving neuron will generate more receptor sites. So we have more information coming out of the neuron and more information being accepted by the next neuron. So essentially, it's like turning a small road into a big highway. If traffic on that road is heavy enough and they have reason to add another couple lanes, and you can fit more cars on it, and those cars can go faster. But how do we build new connections with brain areas that we might not have had connections with before? We've looked at how the cells communicate and how repeated stimulation of some connected cells can strengthen the connections between them, make it easier for them to communicate and faster. But what if we need to build whole new connections? So that's where the dendritic branching comes from, okay? So on this far left diagram, we see the big like blotches in the middle. That's going to be the cell body for a neuron, okay? And each of these kind of looks like a tadpole. These little tails that are coming out are their axons. So when we start, we do not have a whole lot of connectivity. Okay, we have a ton of neurons, and our brain has to determine where they need to be connected to. So the first way that they can connect with certain areas is at the end of these axons, there's actually little cones that can help that axon navigate to a specific place in the brain. So this axon could move over towards a different area of the brain depending on what the brain is experiencing and where it needs to be connected to become efficient. Okay, so first off, our axons can move. Generally, they do this pretty early on in development. Then what we see is that the dendrites start to explode and grow out like a wild forest, and they're reaching out to other neurons that are being activated at the same time that it is. And so we see all this branching begin to occur. And then over here, the branching out has continued. And so essentially, if we were to look at all of this under a microscope, it's such a complicated map of all of these overlapping dendrites and axons that it's really hard to tell one apart from the other. But we can grow new dendrites, and then at the end of those dendrites, we can produce more synapses. Or on the other hand, we can also produce less synapses, which this might be a better illustration of. Okay, so here This is actually an illustration of what the synaptogenesis that happens early on in development. Synaptogenesis is when we just have this explosion right here, 6 months to 2 years of actual synapses. We produce a huge number of synapses and dendrites, and we want a lot so that we can prune away the ones that we don't need and keep the ones that we do based on our experiences. So we see this explosion of synapses, but then our brain wants to be efficient. We don't want connections that we don't need or don't use. We'd love to use those synapses or that electricity or that dendrite somewhere else where we're going to need it. So what it starts to do is it'll prune away. And by prune, we don't literally mean like cutting or killing, but it can kind of absorb that back into the neuron, or it can be recycled through the glial cells that are in the brain, cleaning stuff up. Essentially, it's just going to either put it to good use where we need it, or it's going to make it go away. As we see by 6 years old, after this large synaptogenesis, that we've gotten rid of a lot of these connections and synapses that we didn't need. So what does this look like in terms of normal age-related development? We read about how the brain develops from the back to the front. So the back here is the cerebellum. I consider it the zombie part of the brain. It's responsible for everything that you don't think about. Okay, so if you only had that part of your brain left, you would be a zombie. You could balance and walk enough. You could swallow. You could breathe. You could do all the basic things that keep you alive that you don't think about, but you certainly couldn't do any higher order thinking. And so we're all born with this intact and highly connected, and then throughout the rest of our lives, or the rest of development, at least into our early 20s, we have various things of synaptogenesis, pruning, and then long-term potentiation depending on our experiences. So one thing that is not, one of the major mismatches we have in adolescence is that our frontal lobe, which is responsible for our inhibition and decision making, is not highly connected yet to our temporal lobe that houses our amygdala and our memory. So if we get an impulse here, it's very hard for our frontal lobe to quickly respond and say, hey, whoa, let's think about this, because there's not a whole lot of connectivity there. So one of the things that we're doing in adolescence is developing that connectivity, but it doesn't really finish until we're in our early 20s. This is obviously not an actual map of the brain, but this gives you a good idea of how many connections are going around, and really this is minimized. It would be much more, but this allows you to see individual ones. So I hope that this short explanation and some of the visualizations that I gave you guys here made it a little bit easier or provided some context to understand what we read about in 2 and 3. Jensen did a really good job of explaining all this stuff into plain terms. But sometimes seeing the pictures and understanding how these neurons communicate with each other and build new connections is very important.